chapter 5, you guys, is all about cell reproduction, okay? And the first one here is just basically about how the cell cycle, what it does during its life, kind of like a human lifespan sort of thing, okay? I love this picture because it looks cool, I think, and it's showing cytokinesis when the cell is actually splitting apart. And this is really cool because there's a fiber that has went around, and at one time it was like a big cylinder. And the fiber goes around and it contracts. The fiber gets smaller and tighter and tighter and tighter until it pinches the cell in two. And that part is actually called cytokinesis. Okay, so you should have this down. If you don't have time to get it down. You don't have time to get it down while I'm talking. Please listen to me so you learn a bunch of this right now today. So the first part of this cell cycle that I want to talk about is called gap 1. And this is just when the cell is growing, living, doing its job, doing whatever it functions. So that's called gap 1. And notice it's in the main phase that we call interphase. You guys see this? Interphase. Now the next part is called the S phase, and it's called S for synthesis. You guys, I don't want you copying right now. Pay attention. Austin, stay with me, man, because I'm going to teach it right now. I'll let you do it, copy it later. So the S stands for synthesis, which just means DNA is synthesized. The DNA is being copied. The cell needs to copy its entire instructions, all the DNA, so that when it splits, one half has a set and the other half has a set. Or the two new cells each have their own DNA. Then we go on, and there's one more that is still an interphase. Notice that this whole time period is interphase. Now we have two sets of DNA. So the cell's not doing its job. Right now, it's getting ready to go into becoming two cells. So the next one's called gap 2, and it's got to grow some more. It's got to get bigger so it can split in half. And it's also, with the model here, it needs a bunch more stuff. You got to get more mitochondria, more chloroplast, if it's a plant, more cytoplasm, everything it needs. So it's getting big. Okay, now, the star of this chapter is right here. This is mitosis. And the whole phase, the whole little triangle is known as the M phase. But in reality, mitosis is just these four, and cytokinesis is separate. Mitosis is the time that the cell actually is duplicating, making two nuclei, the nucleus, right? Cytokinesis is what I showed you in the beginning, when the cell is actually split. So that's when the cell membrane, right, split the animal cell. Now, I want you to memorize this right now with PMAT. I don't know if it's going to write for me. P, and write it down. A-T, PMAT. It's just a stupid acronym, but it's going to help you remember the order of mitosis. So, look up here if you have Superman eyes, or look on your paper. What does the P stand for? Prophase. You're right. Many times it's been starting for phosphate. This is just an acronym like UCLA, University, California, Los Angeles, UNR, right? So that somebody just came up with this, PMAT. Prophase, what's the M? Metaphase, then anaphase, and then telophase. And now you know the order of mitosis of copying, not copying, of making two nuclei. Okay? Then, the last thing here you don't have to write down anywhere, I don't think. No, you don't. Because mitosis occurs only if the cell is large enough and the DNA has become damaged. I just want to bring it up to you that along the cell cycle, there are checkpoints. So it gets about here, and maybe it's like, you know what, we need to divide. It gets in here, and it's like, hey, did we copy the DNA right? because if we didn't, we better not finish this off. Okay, so it has checkpoints before it moves on. So that's your cell, what's it called? Cell cycle. Okay, on this one, I want you to copy this down. You should have already done it. So if you haven't, just look for this table. It's in your book. What I want you to remember, though, you guys, is use this, because here's your test question. I'm not going to make you memorize this. The test question is thinking about it. So skin cells, do they have a tough life? Is it easy being a skin cell? No, we got radiation, wind, all sorts of stuff. We hit ourselves, we didn't fight, I don't know. It's painful, and the poor cells don't last very long. Okay? On a good day, or a good 2 weeks, they'll make it that long. Okay? Cells have to divide before they're hurt. They have to replace themselves. If they have a tough life, then they're going to be dividing more often. That makes sense? That's what we mean by rate. How often do they reproduce? Okay? So red blood cells, not so tough. They're just, moving down the blood vessels, picking up some oxygen, dropping off some carbon dioxide, 4 months. Liver cells. I would think maybe I would think livers have a little tougher. They're pulling out poisons, but they're just good at it, I guess. Because that's like over a year, about a year and a half or something, right? Okay, intestines, same thing like skin cells. That internal lining, that's tough. They got stuff going right up against them. But the muscle in the intestine, say, 16 years, right? And nerve cells, what do you guys know about nerve cells? Do they divide? Big long nerves? What about brain cells? Right, they don't even divide after a certain point in our life. Some will, but most won't. So some cells actually never go through the cell division. They just stay in gap 1. So we call it gap 0. It's a little trivial. I just wanted to mention it to you. Okay, now you could have put a variety of things in this box. Check and see if you need to add stuff that I put, don't erase. If you wrote some other interesting stuff, that's fine, but just put it on the bottom. So get these 3 bullets right there if you don't have it done. If you don't have enough, add to it. If you have something totally different, write it on the bottom and arrow it. It's about cell size is limited. It's a 3-bulleted box. We're still on, 5.1. Cells have to be a perfect size. If they're too big, they'll have a big problem. If they're too small, not everything can fit. Is that the cell membrane can get too stretched out, too big, and look at my fake little sphere. Look up here. So here's a little sphere, a softball, okay? And think about this as being the cell membrane, the skin, I guess you can call it, right, on the softball. If we grow it up to a basketball size, what's happened is now it's tougher to get things in and out because we made more volume. Let's follow me on that. So the deal is the surface area does not grow at the same rate that volume does. Just because volume's 3D. Now stay with me because this is kind of abstract. And for your test question, I'm not going to be really making you do the math. I want you to understand what I'm modeling here. Okay? So are you with me? Okay? So the deal is, look at this cube. So look at the math that's already been done. Eyes up here. So I take a 1 centimeter cube, right? So the surface area, length, times the width, times the number of sides equals 6, right? Centimeters squared. Volume, 1 times 1 times 1 equals, so look at the ratio, 6 to 1. You have a higher number here of surface area compared to volume. Just the ratio. Yeah, volume has more space. It's cubed. but the surface area. Okay, now if I double the side of the cube, so now I'm going to go 2. What happens? 2 times 2 times 6, 24, compared to 2 times 2 times 2 is 8. Look at their ratio. They're getting more similar. Where the surface area is going down, whereas compared to volume's not. That means it's harder. You better have more spots, doors, windows, ways to get in and out. Just follow me on this. It's kind of abstract. And then even more, look here. 3 times 3 times 6, boom, right? See, look at that, down to 2 to 1. So it's because of what the cell membrane is doing. Now think about this building that we're in right now. Big rectangle building, right, you guys? Is it difficult to get out all at the same time? We have to go pretty slow. We're legal. It's a time. We're fine. We get out the doors. We're all walking correctly and making our way out. But it does take at least a couple minutes, doesn't it, really? I mean, if we're really hurrying. But I think it probably takes, what, 4 minutes to clear this building? Okay, anyway. What if I cut the building in half? Okay, got Hager to bring in some big equipment. We cut it in half, and then we push this side, I don't know, 100 feet. Build the new walls. Well, just dream with me, will you? Okay? We have 2 buildings. Look at me over here. 2 buildings. Are we going to be able to get people in and out quicker? Why? Okay, because now we have new walls right here that we didn't have before with doors to get in and out. Okay? Cut them in half again. Let's go down to 4 buildings. Can we get in and out easier? So that's all that's happening with the cell. Smaller is better. Not too small, got to be just right. You still got to fit all that stuff in there. Okay? Do you guys remember the lab we did on cell theory? The frog skin cells, the cork cells. Do you remember all the little cells? Were they mostly about the same size? Mostly the plant cell, the plant cell's a little bit bigger. But all the other ones were about the same size. It's because of this. Now you're supposed to circle a box on your form there. And we are moving here. I'm almost done with this section. So, there is a really cool nerve cell, just one cell, that goes from like your spine, in your hip area, all the way down your leg. I'm not sure where it stops. Get extra credit, research for Miss Truth. I want to know where does it stop in our leg? Does it go to our ankle? Does it? Well, look it up. I'm not sure. There's a nerve, one cell, that I know for sure it's going down to the knee. It might be farther. Okay, if I had to guess, I think it does go down to the ankle, but I don't know. Okay, you guys listen. So that means the nerve cell, one cell is long. To be that long, it's going to have to be extremely skinny. And then it's okay with that surface area, right? Because it's elongated like that. So bigger cells have to be elongated, okay, for the same reason. So look at these cells. Even this red blood cell, you guys see this indent? I taught you about that indent in the coloring. It's more surface area. Okay, you smushed it down there, you got a little more surface area. Okay, that's that one. Let's keep moving. Okay, so 5.2, you guys, we're basically going to go through how does it divide. How does it divide? What's the process? Okay, first we have to talk about the DNA, okay? You should have written down the different ways DNA is organized. This is the simplest way, the DNA double helix. This is the model of it right here. It's called a double helix. What's helix mean? Spiraling. Double because it has 2 sides. Okay, see that? 2 sides. So there is your double helix. Now, it's a big long line too. I don't know how long, but you can look that up. Give me some more bonus. I think it's at least a meter or so, your strands of DNA. Okay? They have to be super coiled. There's these special histones, these proteins that help them super coil. Then this chromatin, it's just this strand now supercoiling on itself. And now you could take that strand and make it supercoil right here. If you guys, I want you guys to look at a rope. Look closely at a rope, even yarn. All it is a bunch of twisted little fibers that makes it thicker and thicker. That's what happens until we get to this chromosome. Now this chromosome actually has already went through synthesis. There is 2 sets of DNA here, and it's being held together by this area here. So this is exactly the same as this. That's where all our genes are. Now this page here, I want you real quickly just to do this sketch of the chromosome. You don't have to label the telomeres. Don't worry about that. But label centromere and chromatid. Chromatid. Okay? This one chromatid is an exact copy of this other chromatid. So I try not to draw it like an X. You can if you want, but I just try and draw it where I have 2 separate lines and I get them close together. Because that's really what they are. They're not crossed over each other. Okay, so here is what? What phase is this? Read it. What phase is it of that? It's interphase. You see it? So you need to remember what interphase looks like and what happens, which is what's copied. The DNA is copied, so we call the S phase happens in interphase. Okay, moving on. What's this one called? Prophase. And now we can see the chromosomes. Look right here. Actually, let me go back. You see the real cell of the other one? They don't have it. Darn. We'll see them later. Okay, so chromosomes are starting to really coil on each other. The nucleus is breaking down too. Metaphase, let's memorize it right now. M. The chromosomes line up in the middle. That's what these blue things are. They don't perfectly line up like our little color illustration. What's this one called? I remember this because that reminds me of a capital A for anaphase. But this is kind of cool. Those chromosomes, they move around. They're in fluid. And those spindle fibers attach to that centromere where they're stuck together and pull on it, and then the legs of the exit, they're not even an X anymore, the line. It's just that it pulls from the center so those ends fall behind and are getting dragged. Okay, so that's anaphase. You can see they're red right there. What's this one? And remember, with mitosis, we're talking about the nucleus. We don't even care that this is occurring. What's happening here? The chromosomes now are linear, one set. The nucleus is rebuilding. They're starting to uncoil. Now, we're done with mitosis, we get cytokinesis. And you can see the pinching in. But plant cells, they form a plate. They've got to make a new wall. You'll see that on the movie. Okay, doing great. Let's keep jamming here. Which one's extra credit? 0.4? Okay, so E 0.3. Okay, this is pretty interesting, and if you thought this stuff was boring up to now, I hope you get a little more interested, just because this is going to lead to cancer. And all of us in one way or another know about cancer and have it, affected it differently in our lives, or might in the future. Part of our objective, you guys to be able to make informed decisions, say about treatment choices, and understand what's going on. So let's do internal factors first. Just a teeny little thing for that box. The cell has to be, the cell division, the cell cycle has to be closely regulated, and to determine if the cell should divide or not. So in internal factors, there are special molecules that help with this cycle, help that to occur, PMAD, cytokinesis, and even synthesis. And they're called kinases and cyclins. But I think what's even more interesting is they're actually triggered by the external factors. So what are the external factors? First, they include physical and chemical signals. And then you need to get that stuff down in the external box. Okay, check it out, you guys. So, and you'll see this on the video. I know it's getting long, but stay with me. Let's get it done. Okay? You just take a couple of cells, you take a little piece of tissue, stick it in a petri dish, and many times they'll divide and grow. And when they do, though, when they touch each other or even the sides, they stop. That's an external, physical thing that's helping it, factor. Okay? But cancer cells are not working correctly. So when they hit each other, they don't get the trigger. They don't know, hey, stop it. Stop dividing. Cancer cells also grow too quickly, like we were also talking about. And they never get fully formed. They never do the job they're supposed to be doing. They're too busy dividing to even try and do what they're supposed to be doing. So these are carcinogens. Do you guys see this one? So substances, the definition for carcinogen, a substance is known to promote cancer. And smoking, radiation. Radiation like this could be just from the sun, like this is skin cancer in this picture here. It's melanoma, and it is a deadly cancer. There are some skin cancers that aren't as bad as this one, thankfully. And then there's some viruses. You guys should be getting the HPV virus, right? Human, saying that right? Yeah, human papillo thing. So we do know, we're now recognizing that some viruses are messing with the DNA of cells, and then they can get them to divide. If the cells are dividing more, and that's making more places for the viruses to divide, or not divide, for the viruses to reproduce, it helps the viruses. So we're going to get to that. But yeah, so first what causes it, because you guys can control this. You guys can make educated, well-informed decisions here. Smoking, they're carcinogens. Okay, you're putting burning material, smoke in your lungs. You're smoking now, you need to stop. You need to get as quickly as possible. Or you just realize you're buying in and you're going to go ahead and get cancer eventually. Because that's what happens. Okay, eventually you're going to get it. Some people are lucky and they don't get it, you know, as quickly as others do. Their genes or maybe other choices. Radiation from the sun. You guys need to be using sunscreens, hats, whatever. Okay, cancer cells. Get that one down. You guys are doing great. I know this is getting long, but you're doing great, and we're just going to get her done. So cancer cells, like I said, they do not carry out necessary functions. They are too busy dividing. Where did they come from? Because they're not foreign, you guys. They're that person's cells. So they come from normal cells, but that DNA, the genes in those cells has been mutated. Something's gone wrong. And so it can be inherited too. But for many diseases, if it runs in your family, you want to know your risk factors, you want to know ways to prevent it, and then you can. And thankfully also now cures, you guys were mentioning. So how are we going to get rid of it? Well, we have to radiate or use chemicals. We need to kill those cells that have it. And, you know, there's healthy cells around it or intermixed in it. What if you have blood cancer, for example? So you got to kill a bunch of cells, and hopefully you kill all the cancer cells without killing the poor organism, the poor individual, if it's the human cancer we're talking about. Okay? So it's tough. And all of us have been affected in different ways. Some of us have really close stories, and it sucks. Okay, a little bit more of the details here. Cancer cells, so tumors, you see your tumor box? They're simply disorganized clumps of cells. There's two types of tumors, one's worse than the other. So then you'll go to the benign box. Not benign, you see it under there? So tumors are either benign or malignant. The benign ones, they're the much better ones. Because you can normally just cut these out and remove them. They don't spread. The cancer cells don't spread. Malignant tumors, they can do what we call metastasize, which simply means they can break off the tumor and spread throughout the body, ending up in a different spot where they can start growing there, and then we get more tumors in other areas. Okay, apoptosis. This is normal, and it's programmed cell or death. There's some people have some genes that actually will be in your genes are all in your cells. So it'll actually stop cancer cells. The gene recognize, hey, this is a cancer cell, and it'll kill that cell. But that's fighting cancer. And that's a really cool cure if we can figure out how to get those genes in people that don't have them. And we can. We probably will. But this is just normal, where cells can basically stop themselves from reproducing and potentially even just dying. And you don't need to memorize this, but I think it's cool with this part, so I wanted to share it with you. When you were a little embryo inside your mother, you had webbed fingers, you had webbed toes, you had a tail, a little tail. How did we get rid of those? Apoptosis. The cells, further on in development, they died, those cells died, and then you had gaps there. Now, a lot of us are born with webbed toes, or maybe even webbed hands. Normally people would do surgery, because that, would limit them with the function of their hand. But a lot of people have the little bit of webbing, or maybe just to the first joint, and it's just because it didn't work right. The cells lived a little longer than they should. It's not a big deal. Was there a question, Cole? What? Quickly, because we are going to be done in like 2 minutes if you guys hustle. So the bottom of the pyramid, cells, here we have, develop into mature forms. Cells, bottom of the pyramid. No. Oh, you missed that. Come back here later. Right here. We're going to start at the bottom of the pyramid. We're going to build up. Yes. Develop into their mature forms through the process of cell differentiation. The idea being here, you all came from one cell, sperm and the egg coming together, one cell, but now look at all the different cell types you have. That's called cell differentiation. And you look at these pictures here, skin cells look very different. You can't even see barely. If you got up here, looked on the computer, look in your book, you can almost see the little cells that are in that bone tissue. And then here's little cells inside those intestines. And those are villi, again about surface area. Anyway, this second bullet is extremely important. I'll bet you guys vitamin water that you're going to get some sort of question on your proficiency test on this. They always want to ask this. Do different cells have the same DNA in them? Because your blood cell and your skin cell have the exact same DNA in them. So they have the same genes. Do they have the same genes in each cell, the same DNA? The answer is yes. The thing is, so the second bullet, cells become different because different genes are being expressed. the cell only uses the genes it needs. It ignores the other ones. Okay? But every cell has your exact genetic copy, the exact DNA. So look for that one on the test for me. Okay. These are the other three boxes all in one slide. That's our last slide. We'll come back to SpinFlip another day. I've flooded your brain enough. For what? No, we're not. This thing is bugging me. I need to clean them up and get them out of here. Oh, it's turned on. So you should be aware of this. This is the hierarchy of a multicellular organism. Cells make up what? Tissues. Tissues make up organs. And organs make up systems. Okay? So that's all it is. Now, the test questions and multiple choice sometimes confuse people. They go the wrong direction. You know, I might say, organs are made up of, and what's the answer? Different tissues. Or I could say, groups of organs make up a system. See how you could go either way? So you have to read carefully. All right. You survived. You're going to get up. I'm going to get you up out of those seats. Grab one of these. I have two stacks. Hop up. You've been sitting forever. Grab one of these from that stack or one of these from that stack. Just one each, please. Well, if it's cancer, yeah. And that's not good either. I don't think so. Oh, what did I do?